Okay, so for the next presentation, we have Dr. Florencia Caro, who also hails from Harvard University, and she'll be speaking about the transcriptional silencing by TsrA in the evolution of pathogenic Vibrio cholerae biotypes. Just a reminder for the attendees, feel free to discuss in the chat, but please post your questions in the Q&A tab. With that said, Dr. Caro, take it away.
Thank you. Can you hear me and can you see the cursor?
Yes, we can hear you, we can see you, please proceed.
Perfect. Thank you so much for the invitation. Today I'll be telling you about TsrA, a transcriptional regulator of virulence gene expression in Vibrio cholerae. Vibrio is a gram-negative bacteria that spreads through contaminated food or water and causes this massive diarrhea that has a higher than 60% mortality rate if left untreated. So we know that there's been 7 cholera pandemics in the world, and the first 6 pandemics were caused by this classical Vibrio cholera biotype here in orange that was actually completely replaced in the 1960s by this L-Tor biotype in the 7th ongoing and current pandemic. And the 7th pandemic spread throughout the world in 3 waves, all stemming from the Bay of Bengal, for example, here first to South Africa and then to Peru in wave 1. And here the wave 3 spread that caused the outbreak in Haiti after that earthquake hit the island in 2010. And so in the Macalano's lab here at Harvard Medical School, we use isolates that are representative of each one of these pandemics and or waves to study how this bacteria is so successful at spreading in the globe and causing such severe disease. And so 0395, CC6706, and Haiti H1 are the isolates that I'll be telling you about today. And so as I said, we're trying to understand how a bacterium, how this Vibrio cholerae bacterium can turn from non-toxigenic and non-pandemic to cause very severe disease and spread throughout the globe. And really this property, these 2 properties hinge on 3 factors. One is possessing the O1 antigen that is expressed here on the surface of the bacteria. Only O1 antigen Vibrio cholerae can spread pandemically. The second one is this toxic co-regulated pilus, TCP, that's expressed here on the surface of the bacteria. And without this pilus, Vibrio cholerae cannot colonize the gut of the host. And so it's really a key and essential virulence factor for Vibrio cholerae to cause cholera. And then next is obviously the cholera toxin. So interestingly, TCP serves as the receptor for the cholera toxin phage that, as the name says, encodes for the genes that then produce cholera toxin here in red. And so what you can see is that this TCP pilus and this cholera toxin phage are all genetic islands that Vibrio cholera has acquired through horizontal gene transfer. So these are foreign DNA that Vibrio cholera accepts, and then turn by expression of the genes that are contained in these genetic islands into this very virulent bacterium. And so actually, Vibrio cholera evolution, here I'm depicting the small and the large chromosome of Vibrio cholera. Throughout evolution, these acquisition of horizontally acquired horizontal genetic elements drives the evolution of Vibrio cholera. So for example, in the L-Tor strains, you have the VSP1 Vibrio 7th pandemic island 1 and 2, and then here in the wave 3 strains, this SXT element that encodes actually antibiotic resistance genes. So these genetic islands provide traits that contribute to bacterial fitness and play a major role in bacterial adaptation and genome evolution. But acquiring this foreign DNA, this xenogeneic DNA, is actually a high risk, high gain situation. And the high risk is because if you have new genes that you have to incorporate and into your genome, you must integrate them into this fine-tuned and pre-existing transcriptional program. Otherwise, you will incur a fitness loss. And so bacteria have devised ways to control and the expression from these genetic islands. And so here I'm depicting a phage that's injecting its DNA, this foreign DNA, into the bacteria. And we know that foreign DNA, xenogenic DNA, tends to be AT-rich. And bacteria have these xenogenic silencing proteins that will bind the DNA and silence it. And HNS is a histone-like nucleoid structure and protein, functions precisely as such a xenogenic silencer. So it's a pleiotropic repressor of transcription that recognizes AT-rich DNA and acts on DNA topology in order to repress transcription at this site. HNS in Vibrio cholera regulates about 20% of Vibrio cholera genes. And in particular, these genetic islands that I was just introducing to you that are very important for Vibrio cholera pathogenicity. But additionally, also the type 6 secretion system, which is a system virulence factor that we study here in the Macalanos lab that Vibrio cholera uses to inject toxic protein effectors and kill adjacent cells. And we had seen in the lab that this TsrA protein was functioning very similarly to HNS in that it also repressed genes encoded on these genetic islands, but we didn't know much more about it. And because of these, the important function of these genes, we decided to explore this protein further and try and understand how it works. So the first thing we did is we took these 3 strains that I introduced to you, the classical, representative of the classical and the LTOR biotypes, and we made TsrA mutants in each of them. And the transcriptome analysis of each of these strains. So in this heat map, you have this data summarized, and this is basically a TsrA regulon, the set of genes that are differentially regulated in the presence and absence of TsrA. And in each one of these lines, you have 1 gene, and each one of these columns is 1 biological replicate of 3 of either wild type or the TsrA mutant in the classical or LTOR strains. And yellow means that there are higher mRNA levels, and blue means that there's lower mRNA levels. So I want to bring your attention to the fact that the TsrA regulon really consists of mainly genes encoded on genetic islands here listed on the left. And if you look at, for example, these genetic islands 14, 21, and 23 present in the classicals, but not in the LTOR strains, you can clearly see that in the absence of TsrA, these genes on these islands are upregulated, so mRNA levels are up, indicating that TsrA really acts as a repressor of gene transcription when it is present. And this is true for every gene that TsrA affects. So we can say that TsrA is a repressor of gene expression of genes encoded on genetic islands, much like HNS. So when we compare this regulon to the HNS regulon, we can see that pretty much all the genes that are encompassed, that are present in the TsrA regulon are encompassed within the HNS regulon. So they have indeed similarly and overlapping functions. We had also seen, and so in order to further understand the function of TsrA, we saw that it had some structuring similarity to HNS. And here is a schematic representation of HNS. This is the oligomerization domain on the N-terminus, and this is the DNA binding domain of HNS. And HNS interacts in a head-to-head and tail-to-tail conformation here at sites 1 and sites 2. And there's a crystal structure actually of this oligomerization domain that's been solved from Salmonella typhimurium. And you can see sites 1 and sites 2 here in this crystal structure. I should say that this was solved without the DNA binding domain. Protomers, when you concatenate protomers of this oligomerization domain, what these researchers have shown is that HNS can form this ultra structure, a superhelix that has a 280 angstrom pitch and 180 angstrom diameter. And the model by which HNS is thought to interact with DNA is if here in orange is the oligomerization domain in the middle, and the DNA binding domain is protruding outwardly and and interacting with two AT-rich DNA helices. And in this way, it's affecting DNA topology and supercoiling in order to repress that region as a whole. And so we had seen that we did structural homology modeling of TsrA here in pink and this Salmonella typhimurium HNS oligomerization domain. And as you can see, the structural overlap modeled is pretty much identical. I should say that TsrA is a smaller protein that has no DNA binding domain, at least from what we could predict from the primary amino acid sequence or structural sequence. And the only difference between these two oligomerization domains was this gap of 13 amino acids creating a smaller alpha helix here. And so we modeled this, the shorter TsrA molecule. And if we go to the schematic representation, this would be like saying this oligomerization domain of HNS, a TsrA is exactly the same, but shorter. And so we asked whether we could model the same type of head-to-head and tail-to-tail interaction that we see in HNS with TsrA. And indeed, we can model this site 1 and sites 2. And we can then further ask whether TsrA molecules could form the higher order structure like HNS that's known for HNS. And indeed, we can. And this structural model suggests that TsrA forms a higher order superhelix, but of different proportions to HNS with a shorter pitch and a smaller diameter. Now, without a DNA binding domain, how does TsrA exert its regulatory role on gene expression? So there's two things that we can think about. So TsrA has this N-terminus small domain that could be potentially interacting with other protein partners. But an intriguing possibility that we want to explore is whether TsrA could form heterooligomers with HNS. This would actually be interesting because it would diversify the range of conformations that an HNS superhelix can adopt, making it shorter or longer at different parts of the DNA, thereby affecting gene expression in a different way. We also want to explore the possibility that because now the only difference between these oligomerization domains is the length of the TsrA molecule, perhaps TsrA could function as a molecular wrench that if you increase the length of it, you could actually act and increase the pitch of that superhelix in ways that you can control. And so we would like to play with this and create a molecular adjustable wrench with TsrA. And so with that, I just want to thank the Macalanos lab. Nicole Place is a research assistant that helped with cloning aspects of this project, and Dr. Caro, who helped with a structural homology modeling of this protein. And I'll take questions.
Thank you, Dr. Caro, for a great presentation. Very much appreciated. So I have a couple of questions, and I'll wait for participants in the chat to ask a question as well. But I'll start. In your knockout models that you had of the TsrA knockouts, why can't the HNS just compensate for the loss of that particular complex? Because from my understanding of it, they have overlapping genes that they suppress within the genetic islands. If you lose that particular TsrA gene, can the HNS just compensate for that?
That's an excellent question. If the model that we're predicting is right, that TsrA interacts somehow with HNS, I think you could see either a no effect or a synergistic effect of losing, say, both. I predict that TsrA, that HNS would actually be able to compensate if the model is right. So yeah, but I think a double knockout of HNS and TsrA may be able to address properly what contribution each protein gives to this regulation.
Okay, because I remember in, I think the microRNA data where you have the knockouts, you had an upregulation of those genes. I was surprised to see that because I imagine the HNS is still there. They would also just completely suppress it as well.
Right. Maybe the extent to which the genes are upregulated, right? The magnitude of repression, yeah, is different.
Right, interesting.
Yeah,
okay, cool.
That would be a very interesting, experiment to do.
And then another question, because of the lack of the DNA binding domain in this TsRNA complex, I guess, do you envision experiments where you can do like bioID to identify the protein binding partners that it actually interacts with to identify the whole complex?
Yeah, we are currently making tagged versions of both proteins, of both TsRA and HNS, to see if we can pull down protein binding partners and hopefully identify each other interacting plus other probably interactors. Yes.
That'd be cool.
Yeah, that'd be cool.
Okay.
What happens with co-infection? Sorry to interrupt you, Ashton. What happens with co-infection in terms of the virulence factor? Are there known pathways that can exacerbate the infection with cholera?
Unknown pathways, host specific or cholera specific or Vibrio specific?
Cholera specific.
There's accessory toxins that cholera can express that could exacerbate the disease, but really the signature virulence factor is cholera toxin. In fact, you can take cholera toxin, purified cholera toxin, and put it, administer it to human volunteers and recapitulate fully all disease symptoms, indicating that it's truly cholera toxin that's driving this disease. Vibrio cholera that does not have cholera toxin is actually not called cholera. It's a different disease. It's a diarrheal disease. Yeah. So.
Interesting.
Yeah. Interesting. And then we are running a little bit behind time, but maybe just this one question you can answer from our participant. They're asking why specifically TsrA gene for transcriptional silencing doesn't have any advantages.
So we believe that the advantage of having these types of xenogenic silencers in a bacterium that tends to acquire, it's extremely competent for acquiring exogenous DNA. And so it needs to have these types of silencing proteins in order to first acquire the DNA, shut it down, and then integrate it slowly throughout evolution to the transcriptional program that actually drives virulence in a controlled way. And so I think that TsrA in Vibrio is an important factor, much like HNS, to drive this kind of, the transcriptional program,
Okay, that makes sense. Thank you so much, Dr. Caro.